Hey, greetings from the Morgan field. Here. Hope everyone's quarantine is going as well as can be expected. As you can see, we're all past due for trim. At least we are here. There. So a few of you guys have turned in your materials. That's great. I can be flexible with due dates here. Just get them in if you haven't already done so. Okay. Talked about genetics, Excel reproduction, everything last time. We have kind of a short session this time. I had hoped to have a full, whatever it's called, collaborator, I believe it's called, there on Blackboard. I looked at that. Apparently, my video camera does not agree with Blackboard, and there's technical issues, and I have something of a Luddite, so, whatever. Anyway, okay. We're going to take a look today. It's going to be sort of a short session today. We're going to look at chapter 10 and just a little bit of chapter 13 today. Okay. I'm going to cover the positives and negatives and some of the practical applications of biotechnology. And we're also going to get into viruses, and then you get to hear a rant about vaccines. Okay. So if you would, let's look on, we'll start on 174. And it's talking about personal genetic testing. And, this is just one of several aspects of genetics, biotechnology. Excuse me. And what you can do there is they can take a sample, your DNA, they can analyze as what you have. They will tell you the risks that you are susceptible to as far as what diseases you do have or what diseases and disorders you could potentially pass on to any offspring. A lot of times, a person and their partner will go in and they'll be tested, and they'll get genetic counseling to see whether it would be advisable to have children or not. Okay, and we're not going to go into a lot of detail on this stuff up here. It's a little technical for what we're doing, but it is in there if you need it. I want you guys to understand the basic concept. Okay, process called recombinant DNA. Okay, what you can do is you can use an enzyme. Remember, that's a protein in there, which speeds up a reaction, and you can use what's called a restriction enzyme, and that will cut a segment of DNA off of there, or it will actually it will isolate a gene. It will isolate a given gene from the strand of DNA. You can cut that DNA out, and you can actually plop it down onto another petri dish, and you can actually make copies of it. Okay, you can use that, and you can also take that gene and you can plug it into another DNA sequence, and this can give you some really interesting results. Okay, this has led to some things. It's led to things like pesticide resistance. It's there. It has led to pest-resistant crops. Up there. It's also led to things like, say, glow-in-the-dark Christmas trees. What they'll do is they'll take the DNA from a firefly. They'll find the gene for bioluminescence. And they will plug that DNA into the gene for a spruce tree, the DNA of a spruce tree. And what they will do is they'll plug that bioluminescence in there, so you will get a Christmas tree that actually glows in the dark. Okay. So you can take this there for any number of concepts. I'm going to talk a little bit more about genetically modified organisms here in just a moment. Okay. Let's see. Okay. Often, and there you got to find a way to introduce a gene into an organism, so you can use a virus. We'll talk about that here in a little bit. Or you can use a bit of bacterial DNA. Anything that you use is called a vector. It transmits whatever you're trying to send from an organism to the next. And a lot of times you can use as a vector either a virus or a bacterium. Bacteria are there. You look at bottom of page 176. There'll be a ring of DNA that is called a plasmid. And that plasmid, it can go there, or you put the bacteria into the organism and where you want to introduce the gene in question. That plasmid will get passed on and it'll kind of combine it. Okay? Okay. Yeah, let's look on 178. Okay? And you can see there the breakdown out of DNA, the genetic sequencing. Okay? What you do there is take a sample of DNA, and if I recall correctly, none of you guys are taking the lab this time, which is unfortunate there because you're getting to the point in the semester where if you had been taking lab before all this virus stuff came along, if you had been taking lab, you'd be about to the point where you'd be isolating DNA. And you can do it at home with materials you have at home. If you have laundry detergent, liquid laundry detergent, and you have everything you need, you can look up online how to do that. I can probably be persuaded to post a link there if anyone wants, but you can use laundry detergent. You can actually isolate DNA, okay? And it has the consistency of snot, okay? And I didn't believe that when my professor told me that, but it really does have the consistency of thick snot. And so what you do there, if you want to sequence DNA, like you see there, or on top up left up to 178, okay, is you can get your DNA, and you can smear it out on this gel. This chemical gel. You run an electrical current through it. And that is a process called gel electrophoresis. Okay. And what that will do is that will break in the different genes. It'll break them down under their component parts. And what you'll see, get this out here. What you'll see is something like this. Okay, you will see a sequence of DNA, and you'll see things broken off. You'll see these very distinct bands, and they'll look. Let's see if we can get this here. They'll look something like that. Okay, they'll be spread out in bands, kind of like that. All right, and this is really useful here. Or whenever you get into your CSI type shows or your DNA evidence and whatnot, this is how they'll go about it. What they'll do is they'll have a suspect. Well, they'll have the DNA taken from the perpetrator, and they'll run it through a gel electrophoresis process. It takes about 24 hours. Okay, it's not like on TV where they get instantaneous results. It takes about 24 hours to run the process. And what they will do is they will run it through, and they'll get, okay, here's the DNA found at the crime scene. And when they take the DNA from the suspect, if it looks like this, and there, okay, and then here, if you have a good match like that, okay, where all the band ends show up at the same spot and everything, there's a pretty good bet that your suspect is in fact the one that did it, because it's a match up there. And they'll narrow it down to, one and however many million or billion. And you guys bear in mind too, the population of the planet Earth is about 7.5 billion, give or take. So if a 1 in a million chance, there's still 7,500 people that could have done it. So you want better odds than 1 in a million. Now, if they say something 1 in 10 billion, and there's only 7.5 billion people left, well, there's your guy. So DNA evidence can, and your lawyers are always going to sit there and tell you that, well, they never conclusively show that the guy did do it, excuse me. But it can conclusively show, however, that they did not do it. Let's say you've got this circumstance here. Okay, now we've got a band here from the suspect that was not found at the crime scene. That guy didn't do it. He's clear. Okay. So, if you're ever on a jury, there, just understand these things. Now, your lawyers, people, the lawyers for the defense are going to be banking on the idea that, hey, no one understands this stuff. Okay, so they're going to try to obfuscate. And the prosecution, they figure no one understands this stuff either. So, just have a basic idea how these things work here. Okay, keep in mind of your numbers. Okay, and let's look up here on top of 179. Okay, guys, our DNA is pretty similar to DNA pretty much any other organism out there. In fact, our nearest relative, of course, is chimpanzee, 99.9% the same. Okay, you see the us and the pufferfish. Okay, just a few small changes. Okay, you see in some cases the exact same sequences are found. For some of them, just minor variations. So, guys, again, remember, we talked about how just a couple of nucleotides or a couple of nitrogenous bases difference can make all the difference there. Well, there you see it again. Okay, so the sequencing there. So it's these small differences, even out of millions upon millions of base pairs, can make all the difference there. Now, let's see. Okay, and genetic sequencing, of course. This here, it's a major thing. People are always wanting to make some controversy about it. They'll citing privacy concerns. They will cite, the idea of possibly being able to patent a gene, which is a little questionable there. How you can patent something that has always existed, but that's here or there. And about the year 2000, Human Genome Project that was completed. And the genome, guys, GENOME, that is just the sum total of an organism's genetic makeup. Okay, we sequenced the human genome in the year 2000. I was in grad school for biology, and it was a very exciting time. And there, they figured out what every identified gene in the human body it does. What sequence there, what chromosome it's found on. They've identified all of that. Now, I believe we spoke previously about the concept of junk DNA. There's a large quantity of DNA in our cells that, as near as we can tell, has no purpose. Okay, it's still an open question as to whether or not it serves a purpose. If it does serve a purpose, what purpose does it serve? Okay, but we have sequenced all the genes in the human body. Okay, the study of genomes, that's genomics. Okay, DNA profiling. We just talked about that. Okay, let's see. All right. Genetic engineering, genetic modification. Okay, a lot of controversy. Okay, and a lot of that controversy, guys, probably 3/4 of the controversy there is simply that people don't know what they're talking about. Okay, people hear, oh my God, it's genetically modified. Oh no. How? Okay, but what happens there is, guys, we have been modifying genes in our plants or in our animals that we're raising as livestock or whatever. We have been modifying them since we've been growing plants and raising animals. Okay, everything's genetically modified. The vegetable that we know is corn, which is actually a fruit, but we think of just vegetable. There, or the corn. What we know as corn is very, very different an organism than what it was as there in the time of the Mayan Empire. Okay, and you can Google that. You can see the difference. Okay, that's due entirely to genetic modification. What a farmer did is they say, okay, I want this ear to be a little bit longer. I want it to produce bigger ears. There. Okay, I've got my bigger ears. Tastes kind of like crap. So let's take this ear corn over here, and we're going to cross it with this big ear here. So this one here, it's a very sweet corn. We're going to cross them. We're going to see what happens there. And people will just go on with that. Okay, genetic modification has happened for millennia. Okay, the only difference now is that instead of doing it in terms of one generation or the next there, one generation to the next there, crossbreeding, we're doing it with a little more high-tech technology. Okay, we're going in and taking genes from different organisms. Okay, and if you look on, page 182, you can see there's a mold-resistant corn up there. You can get some that will glow in the dark right there. Also produce various chemicals that produce hormones for one thing or another. They can be modified to increase the yield. You can Google golden rice. That's a rice that's been put into place in areas that don't have the greatest food security. It gets twice the nutritional value of regular rice. It's there. So overall, guys, this is my scientific opinion. It's shared by most people in the scientific community, is that genetic modification is safe. Okay? It is a good thing. It is safe as far as we know. Now, my wife's in here sitting in and trying not to heckle me. I'm sure she's going to be giving me an earful when I log off here, but that's fine. And she's giving me a look right now. Now, she's giving me a bigger look now. But the caveat that I'm going to make for it on that is that genetically modified organisms, okay, maybe they are safe. They most likely are, but at the same time, I have no issue with some sort of notice that it has been genetically modified. That way, the consumer can make an informed decision. Okay, and the only reason I say that, I fully support GMO organisms pending further evidence. Okay, like everything else in science, pending further evidence. 1950s, there was a medication. It worked wonders. Okay, women and pregnant women loved it. Doctors prescribed it. They loved the effects it had. Medication called thalidomide, and it was great. It worked exactly as intended. Okay, morning sickness. Took away morning sickness. No more morning sickness. Okay, it was the greatest thing. Until the babies came out, a few months later without any arms or legs. Okay, it caused, it was what we call a teratogen. It caused birth defects. And it took them a while to figure this out. Okay, so sometimes, guys, you have unintended consequences that aren't immediately clear. So I'm fully in favor of GMOs. I personally believe they're safe. Most people in the scientific community will tell you that they're safe. You got a few that disagree, but the general consensus is that they are safe. I agree with that. Now, with that said, and again, in there, I have no issue with warning labels. Okay. Overall, I think the benefit that outweighs whatever harm they may be. Okay. Like I said, it's kind of a short class here. We got our last section on this video, then I'm going to make another one. Okay. If you look on 184, 185, talks about modifying humans. Okay. And of course, this is one of our kind of moral gray areas here. Is it okay to be tinkering around with humans, and if so, how much? Okay. Gene therapy. Okay, they can introduce intravenously into your cells. They can introduce a gene in there that, just for example, will correct your eyesight. Okay, you there? This kid in here, you see at the top of 184, he's undergone gene therapy, had immune system, he's been permanently repaired. So they can go in and fix that. Little girl on the second picture there, she underwent gene therapy to fight leukemia. So it does have a plus side. Okay. Again, unintended consequences could arise at a later date, so, caveat emptor, but, again, they're most likely safe. Now, beware, though, because you see on page 185, the concept of eugenics. Okay? This is sort of the end point there of, various isms there. And we saw the logical conclusion of eugenics the 1930s and 1940s with the Nazis. And I hopefully don't need to tell you how that went. The idea of eugenics, okay, is basically either using in the genetic makeup of an individual to determine, what's best is for the human race as a whole. Okay, the idea that some genes are better than others, or some genetic characteristics are better. Okay, and 1920s and 30s, when this was really in vogue, you'd see this. You would see at the county or state fair, or you would see, next to the best cow or the best hog, whatever, you'd see the fittest family. Okay, and of course, people would take this idea there. They would say that, okay, certain people should not be allowed to reproduce because their genes are inferior. Okay, and they would say this with, okay, maybe they've got, some sort of genetic condition, and maybe they've got diabetes, maybe heart disease runs in their family. Maybe they shouldn't reproduce. And of course, this got, elaborated on or extended there. You'd see people being told that, oh, well, you've got a mental deficiency, or you're prone to criminality, or things like that. And, places like the American South out there, are certain other characteristics where, of course, are deemed undesirable. Okay, basically, if you were black, if you, were considered less, and you would be told you wouldn't get to reproduce. And there were forced sterilizations based on race and criminal history, mental capacity, things like that. Okay, so that's the downside there of all this. So you have to be vigilant against. Okay, all right. That is the biotechnology lecture, kind of short and sweet, but we got the basics in there. Any questions, please feel free to contact me. I'll see you back here in a minute there with viruses and the vaccinations. All right, see you in a bit.